The situation that stands as of today is that since the Zoom meeting can only be carried on for 40 minutes time, maybe that we all have to break in that minute and we can just rejoin the meeting and we will start over the class. We are trying to fix this. So since I do not have institutional access for any platform that allows meeting without a time limit, as well as allows recordings for the sessions. And sometimes the offline recordings fail, as it was for the last class. So we had to switch from Google Meet. Let's see how we resolve this for our future classes. But for today's class, we might want to break in 40 minutes time, so maybe 30 minutes from now, and we can just quickly join back. Since it's just you today, there's going to be a lot of me asking you things, maybe. Could you? Okay.
I will be waiting for that.
Okay. So, yes, the last time you left me with two questions. One was regarding the cell adhesion molecules, right? So, okay, I checked that. So, for your your cell,
In progress. Progress. Fix.
so the cells of your the endothelial cells, they are indeed expressing the ICAM molecules, as was said. And apart from that, the question was about CD40L molecule, right? So there, just a second. Let's try presenting the screen. So today's class, we have a lot of information that is going to be there. Gradually, our classes are going to be more information centric because immunology has a lot of details to the subject, right? So we'll just come to CD40 molecule that we were talking about. Just give me 1 second. Hmm. So I'll just answer your question about the CD40 thing that was asked. So CD40L. So I said that anything that is with CD40, anything that has to do with CD, it is associated to the lymphocytes. But it must be also CD40L might also just be on the lymphocyte. But turns out that's not the case. So CD40L, this L stands for ligand, right? So the receptor CD40 is indeed present on the uh, T cells, right? The CD4 plus T cells. And the CD40, which is present alongside the TCR, it is going to interact with the CD40 ligand molecule, which is present on the dendritic cells. So dendritic cells, which is an antigen presenting cell, if you remember. So CD40L, I think the question was where is the CD40L molecule present? So it is present on the antigen presenting dendritic cells. And CD40 is what was present on the CD4 positive T cells. Okay, if this is clear, we have maths. So with this, we can start with today's class. Today we are going to discuss, so we left our discussion around the cells of the immune system. I hope you all could access the lecture notes. So there's also there's also a offline session that was recorded again for the previous class, which was uploaded to the YouTube link. And over there, we added, while I was taking that offline session, I also added certain markers and all that are present on each cell type, right? So all those details, it's just certain theoretical details at what is the percentage for each cell type, which all molecules might be present inside the granules present in the cells. So all those little details which might be of importance, they have been added to the notes, and as it is, the entire note has been shared, uploaded onto the Google Drive. So while we finished, we stopped our discussion at the cells of the immune system. What we move on to today are the organs of the immune system. So if you have
Excuse
a
me,
quick,
ma'am.
yes.
When was the offline session held?
So, but your offline session is nothing. So the live session that you had attended the last Tuesday, so the recording for the session actually failed, right? So I was asked to upload, just do the entire class once again, record it offline, which means like while none of you were present, and have uploaded that link, that particular recordings link to the YouTube. So if you have access to the weekly log sheet, there's a link to that session, right?
Yes, ma'am, I have gone through that video, so I have come across that you have recorded the another lecture, and then at the end, my doubts and someone else was coming
Yes,
there.
yes. I because once I switched to the main screen while I was taking the doubts, I realized that the recording had stopped. So I just quickly started the recording at that point. Mhm.
Okay, ma'am. And ma'am, CD40L, what you have told is present on the dendritic cell, but when the answer key has been came of that
Mhm.
assignment, it is showing me that the correct answer is the helper T cells.
CD40 is present on helper T cell, but CD40L is present on the dendritic cells. If the answers are not aligning to this fact, then there is a mistake in it. Can you
Ma'am,
please
but
tell me the
it,
class, the weekly assignment, or if you can read out the entire question once again?
sure, ma'am, I will be reading the entire question. CD40L is present on the surface of, and the options are B cells, helper T cells,
Hmm.
cytotoxic T cells, and macrophages.
So it's helper T cells, B cells, macrophages, and
Cytotoxic T cells.
CD40L is present on the dendritic cell, which is a APC. So I might just I may buy macrophage to be a correct option if I have to pick one, but it's not the T helper cells. If that's not the answer, then there is other solutions released for the way.
Yes, ma'am, the PDF has been uploaded, but I think that there is no such explanation is given of this question answer. Only the option has been written there.
I'll again go by it, maybe from the updated versions of the book, but something that wrong is not going to be there even on the older editions for the book. It has to be CD40, which is present on T helper cells, and CD40L, which is on antigen presenting cell, which that too the dendritic cells. Okay, I can check that once again if you say the answers are so, and if it's still not aligning, I can get back with NPTL and we can look into the matter, right?
Okay, ma'am.
Bec-
Ma'am, once you have cross-checked this question, then can you please re-explain me that?
Sure, sure. If it is so that the answer is indeed the helper T cells, whatever is the logic by which they are stating it to be so, that we can, if I understand their logic and if that's true, then I'll also take that up. Not a problem.
Okay, ma'am.
Okay. So with this, if everybody is on the same page, we can move ahead onto our discussion of the organs of the immune system, right? So since it was a lot of facts and I do not like want to miss anything out of my memory while the class is being hosted, I have also tried to jot everything down in the notes and we will just be going through it one by one. Once we are done with our discussion of the organs of the immune system, we will better understand the make of the B cell receptors and the T cell receptors for today's class, right? Okay. So, when you look at the entire body, the entire body of ours, you would see that there are multiple lymphoid organs that have been marked, right? Some are lymphoid organs, some are the veins and venules that are supplying to the lymphoid organs, and you will also see, so over here there's another duct over here. So these are everything that is actually feeding into the lymphoid system. Everything has been mentioned in this one diagram, right? So we'll just keep looking at each of them one by one. If I have to classify all these structures, all these organs and different things present in this diagram, they can be classified three um, into three categories. So we first have the primary lymphoid organs, or the we can also call it as the central lymphoid organ, because those are the main place where the maturation of lymphocytes is taking place, right? So we have bone marrow and thymus falling under the category of primary lymphoid organ. So we have read about the hematopoietic stem cells, which are essentially present inside the bone marrow. Once the B cell, the maturation part of the B cell is also taking place completely inside the bone marrow. But once the T cells have formed, they migrate into the thymus, and the maturation of T cells is happening inside the thymus. Okay, so then once the mature cells have formed inside the primary lymphoid organs, these cells then move into the secondary lymphoid organs, which are also called as the peripheral organs. So peripheral because that's more towards it's more dispersed along the entire body periphery, right? So that's how the name has come as peripheral organ, and it is also the site where the antigen and the lymphocyte encounter can actually happen. So what your peripheral fluid that is actually circulating through the body, that carries your antigen and it into these secondary lymphoid organ, where they get a chance to interact with the lymphocyte and they are activated. So under your secondary lymphoid organ, we have the lymph node, spleen, and the mucosa associated lymphoid tissue. If you remember from the first class, we have already discussed MALT. So MALT is where we read it in the context of innate immunity, right? The physical barriers that are there, malt is also a part of it. But now we are saying that it is a lymphoid organ, which means it is also harboring certain lymphocytes. So we'll look into that. What are lymphocytes? How organized are their structures? How they are arranged in the tissue? All those things we will discuss. Lastly, we have the tertiary lymphoid organ, wherein the number of lymphocytes present is very few, but if there is a antigenic challenge, it can import the lymphoid cells and mount a inflammatory response. So as we were saying, so skin is actually again an example of a lymphoid organ, tertiary lymphoid organ. So skin comes under cutaneous associated lymphoid tissue. So you might come across some question which can ask you maybe something like that, which of the following is not a secondary lymphoid tissue? And if you have the options of malt, galt, uh, balt, or galt, so these are different names. So galt is gut associated. We have bronchi associated, we have malt, which is mucosa associated, which in a way comprise these two. So malt is a broader term. Galt and balt is subconstituted constitution of the malt. Or you can also come across galt. So you must know. Okay, so we already have the meeting. So I just request you to so I would request you to please join the meeting in another 10 minutes time, because the meeting might just get timed out. By the next class, we will have a fix for this problem. Don't worry. So CALT is the right option because it is not a secondary lymphoid organ, it's a tertiary lymphoid organ. If you remember the word cutaneous, you will automatically be associating the surface structures along with it. So the skin is an example. And skin, again, we have discussed it in the context of innate immune system. So in the skin, so I'll just quickly finish with this tertiary part because then we will in detail discuss the primary and the secondary lymphoid organ. So while we were discussing about skin, we understood that the skin has a epidermis layer, which is the outermost layer of the skin, and it also has a underlying dermal layer, right? So in the epidermis only, we have certain cells, certain specialized epithelial cells, which are called the keratinocytes. So keratinocytes are epithelial cells, right? And we talked that the class 2 MHC molecule are only expressed by the antigen presenting cells. But over here, there is an epithelial cell called keratinocytes in the skin, which can be induced to express the class 2 MHC molecule like the antigen presenting cells. So it's a specialized epithelial cell. And once it is expressing a MHC molecule, it can also express some antigen complexed with it. And if it is being recognized by certain other cells of the immune system, there might be a secretion of cytokines. So the keratinocyte in itself can also secrete cytokines and mediate some local inflammatory response. But again, since it is a tertiary lymphoid organ, you must remember that that is not something that is usual for the cells in this keratinocyte in the skin to do. It's not something that is always there. It is a specialized condition wherein maybe there's a breach in the skin that is triggering the keratinocytes to assume these functions. Apart from that, we also have Langerhans cells. It's another type of cell which is also present in your epidermal layer. So Langerhans cells is nothing but a dendritic cell that we saw coming from both the lymphoid as well as the myeloid progenitor cell. Archana, are you trying to say something? Okay, okay. So Langerhans cell is a dendritic cell which is there in the epidermal layer. Some cells among the cells of the epidermis, we somewhere have the Langerhans cells present about. So if there is an antigen coming, again, it can actually take up that antigen, process it, and present it along with the class 2 MHC molecules associated to the Langerhans cells. Okay, apart from that, there is some lymphocytes called as the intraepidermal lymphocytes. Intraepidermal lymphocytes, which are so it is having lymphocytes in its name. So you can remember that any lymphocyte has a CD molecule associated to it. So if there is a lymphocyte, there must be certain CD molecule associated to it. And what do the the CD associated lymphocytes can be having? They have certain sort of receptors, right? So intraepidermal lymphocytes, they also have a receptor, which is known as the gamma del type of T cell receptor. Again, it's a piece of information. I'll just give you a quick explanation there. We will look into the T cell receptors towards the end of today's class. But if you visualize, see, this is a T cell, and somewhat the T cells look like this. So we'll look at them, right? This is how a TCR looks. So if you see that we have two such molecules associated to each other, we were so far visualizing TCRs as these molecules or something like this, right? But if you look at the structural feature of it and you make a schematic out of it, this is how a TCR looks. So of these two chains, which are together constituting the TCR, One chain is having a is designated as the alpha chain, and the other is designated as a beta chain. So this is a TCR, a T cell receptor. But intraepidermal lymphocytes, which are also present in the epidermis, instead of having a alpha and a beta chain associated, which is the part of a TCR, they have what is known as the gamma and del chain. So this is gamma del T cell. So everything is like the T cell. It's because it has a TCR, just that these two chains, instead of being alpha and beta, it's gamma and del. So the exact function of why such cell, what is the function of these gamma del T cells, this is so far not understood, right? But we do have some intraepidermal lymphocytes which have this sort of receptor make. Along with that, we also have very few CD8 positive cytotoxic T cells. The normal CD8 positive cytotoxic T cells. Okay. So with that, in the so this is so these all these three cells that we discussed so far, these can be present in your epidermal layer. Inside the dermis, we can have some scattered population of T helper cells, cytotoxic T cells, some macrophages, but mostly these cells have actually are the cells that have migrated into the dermal layer. If you remember that we have seen a diagram where there is a cut made on the layers of the skin, and through that we have certain lymphocytes that can actually extravasate into the tissue, right? So the scattered population of CD4, CD8 and macrophage cells that are usually found inside the dermal layer, those are mostly the previously activated memory cells that have come into place to maybe take care of some antigenic challenge that has happened. So this is so the tertiary lymphoid organs, they no doubt they are important, but the discussion over it is very brief. So it is something is assuming that function because of some requirement of the body, right? I would ask you all to please quickly join back, right? I'm just ending the meeting. You just quickly join back and we will see how we can fix it in the meantime, because otherwise the meeting is going to automatically get timed out in a minute. Okay, I am ending the meeting. Please join back with the same link. Okay. So, as we have discussed tertiary lymphoid organ, we'll now uh, go back to understanding of the more important, the primary and the secondary lymphoid organ. So, beginning our discussion with the primary lymphoid organ, we have two primary lymphoid organs, the thymus and the bone marrow, of which thymus in particular is a little more it has a lot more of detail. Bone marrow is pretty straightforward comparatively. So if you understand thymus, uh, it's a small organ, it's a bilobed organ which is located right above your heart. So it somewhat looks like this. So there are two lobes over here. One of these lobes have been demonstrated, right? So if you assume a 3D ball placed like this and you make a cut of it into the half, right? So this kind of half is known as the transverse section, and if you're going to cut it from the middle, this is known as the cross section, right? So what you see on the screen, this essentially is the cross section of the thymus. So you have two lobes here. Someone has taken this lobe off, and this portion, this hollow, is being is represented in the schematic. So if you're seeing, say, just imagine it, say two Costco balls together. You have taken one Costco ball out, and the other ball also you have cut it in half. So what you see is a cross section. If you see this cross section carefully, what the first thing that you would notice if you go from the top to the bottom, that there is a capsule. So outermost, there is outermost covering called the capsule, which is going to extend certain projections into the interior of the organ. So this is known as the trabeculi. So these are just some names that you must know. So we have the trabeculi, which is dividing your entire organ into certain sections. Right beneath the capsule, this region, so this is the capsule in red color. Right beneath this region, if you see this region, which is more densely packed with cells, this region is known as the cortex. Cortex is the region that harbors a lot of immature thymocytes in the interior part. So this is by normal convention, anything that is on the outside is called the cortex, and anything that is more central of the organ, that is known as the medulla. So inside of the cortex, this region which appears to be less densely packed with cells, this is known as the medulla, which although has lesser number of cells, but over here the lymphocytes, they are the mature lymphocytes. In addition to, just remember that which section of which organ, which particular region of the organ has a more, say, which type of cell is present in which part of the organs that we discuss in today's class. So those are the interest areas that is usually being asked in question answers, maybe assignments, maybe somewhere else, right? So cortex, you have to remember, it is where the immature thymocytes are present, and mature thymocytes are located towards the medulla. Now, if you see that what all cells are present, so thymocytes, if you remember, so why we are calling it thymocytes? Because it's just the T cells that are present, thymocytes, which are developing inside the thymus. So you would not see a B cell is not very likely to be found inside the thymus, right? Apart from the thymocytes, you would see cells called as the nurse cells, and as the name suggests, the purpose of those cells is to provide nourishment. We will discuss this. We also have certain cortical epithelial cells, interdigiting dendritic cells, and some medullary epithelial cells. So the epithelial cells that are present in the cortex regions have been named as cortical epithelial cells. In the medullary region, they have been they are named as the medullary epithelial cells. You would also see some macrophages here, and obviously, if it's an organ, it needs some blood vessel supply for the overall survival and maintenance of the organ, right? So these extra cells that you see, the nerve cells, the thymic epithelial cells, which might be the cortical medullary cells, or the dendritic cells, the macrophages, these cells form a very intricate network. Charvani, I was trying to join you since very long. I don't know why you were not getting connected. Anyway, so we just started, moved our discussion with thymus, and this is the diagram wherein you see multiple cells packed in together. The outer region is being called the cortex, which has immature thymocytes, and the medullary region having more mature thymocytes. Apart from thymocytes, we have certain additional cells, nerve cells, cortical epithelial cells, dendritic cells, and macrophages, which have a very important role to play. So what happens, these extra cells, apart from thymocytes, they together form a very close network-like structure. So if you see that they are all they are all packed very close with each other, like a very, very dense network. This network is called the stromal cell network. So stroma is got something to do with matrix. So anything extra, this apart from the cell, whatever is binding all these cells together, that is known as the stroma. The cell network in itself is binding everything together. So it is named as the stromal cell network. This stromal cell network is actually constituting the entire bulk of the organ. It is providing the entire framework of the organ. So it is something that is binding everything together so that it's not loose and unbound and moves around like that. Okay. So going by, so there are lobules which is separated by the connective tissue strand called trabeculi. We have the cortex with immature thymocytes. The outer cortex also has nurse cells. So these nurse cells, if you see, it's quite an elaborate kind of cell. And if you look here, you would also see that a lot of thymocytes have actually been completely entrapped by these processes of the nurse cells. So one single nurse cell can almost cater to 50 thymocytes, and they take care of the development of the thymocyte, the overall nourishment part of it. You would also see that there are certain dotted structures which has been demonstrated as a dead cell. So in the cortex, there is a lot of cell proliferation happening. The cells are undergoing maturation and proliferation, cell division, increasing the number of cells is also a part of this process. Why? We will understand there is something called as selection that is happening. So anything that is not getting selected, they are taken to death. So there we also see a lot of dead cells in the cortical region of thymus. The nerve cell catering to nourishment of the cells that are important to the body, and ultimately they get a chance to interact with the cells of the stromal cell network. So what do the cells in the stromal cell network do? They are nothing but antigen presenting cells. So if you remember from our previous discussion, T cells cannot recognize the antigens separately. They need the antigens to be processed and presented with the either the class 1 MHC or the class 2 MHC molecule, right? So which inside this thymus, during the process of maturation, they have to meet the antigen so that they can completely become so this is so that they can become immunocompetent, which means they can elicit a immune response. So not that it is eliciting one, but if required, it can recognize the antigen and mount the immune response. So these cells which have the antigen presenting molecule, which is the antigen presenting cells, they have MHC molecules, and they during the process of maturation are going to present different antigens on purpose to the thymocytes. Whichever thymo, so say you have, so maybe you have this one antigen present in complex with the MHC molecule of a APC antigen presenting cell, and you have a thymic cell which has a T cell receptor which can recognize this antigen. So this T cell is selected for, and it is now known to the system that this can recognize this antigen. It is immunocompetent. And whenever this cell reaches the circulation, it is only going to interact with this particular antigen complexed with the MHC molecule. Now consider the case where you have another T cell. So we have another T cell here which cannot bind this antigen. So this T cell receptor, whatever is the receptor diversity that has been generated, it fails to recognize this antigen. So this process, if it is not recognizing this, maybe there is another APC which is presenting some other antigen which this T cell can respond to. This is also selected for them. But if we have multiple other APCs and none of the T cells this can react to or recognize, this cell is triggered for death. So this process, so here we have considered one process where antigen recognition is given priority. Apart from that, if you have a T cell which can bind to an antigen, which is not even being presented by a MHC molecule, such T cells are also triggered for death. So if you look here, thymocytes that react to the antigen alone, they are taken for death. Thymocytes that react to self-antigen. So as I was saying, the body is actually, so we read about self-tolerance. The body actually wants all the different diverse receptors on the T cell to interact with every antigen that is also present inside our body. So, purposefully, every antigen of your body is also being presented to the thymocyte while they are developing. Any thymocyte which is reactive towards your cell antigen complex to a MHC molecule is also taken for death. So thymocytes that can itself bind an antigen is taken for death. Thymocytes reactive to self antigen MHC complex is taken for death. Only the thymocytes that react to foreign antigen complexed with the MHC molecule, they get a chance to reach your secondary lymphoid organ because the body knows that this particular cell is immunocompetent, which means it can elicit an immune response only against the foreign antigen, that too if it is complexed with MHC molecule. So body says that this is a safe cell and this is an immunocompetent cell. Let us ask it to reach the secondary lymphoid organ where it is going to survey, wait for some antigen. If it encounters one, it can mount an immune response. Right? So this is essentially, so who all are presenting these things to the thymocyte? It is all the cells of stromal cell network. Most importantly, it is your interdigitating dendritic cell, which has the primary role in presenting these antigens to the developing thymocytes. Okay, another just a fact about thymus, because we need to understand the organ as well. So once you are born, your thymus is around 70 grams. By the time you reach puberty, it is accumulating some fat. Every organ has a tendency to accumulate fat as the individual grows. So your size is increasing and it is most developed when the person is at puberty. So you are best protected against any sort of infection during the puberty age, right? Through your age, as you say, enter your late 20s, the early 30s, and gradually your thymus, the entire mass of your thymus is decreasing. So this decrease in older age is the prime cause because of which elderly are more susceptible to to compromised immune system. So there we say that their immunity is not so strong anymore. That is partly because the thymus has shrinked. And how do we know that thymus is that important? How people have actually come to know that thymus is an organ that is catering to the T cell development and all those things. So people have performed simple experiments where they have maybe taken a mice and they have performed thymectomy. Anything that has tummy, that means it has been cut off. Surgical removal, anything that has been surgically removed, it has the uh, suffix of ectomy. So thymectomy is a condition where the thymus have been surgically removed. And this situation is also somewhat mimicked by a diseased condition that exists in human, the Dijor's syndrome. In this particular syndrome, your thymus is not well developed. So it's not like there's no thymus, but your thymus is very, very poorly developed, such that your T lymphocytes, the number of T lymphocytes is very, very reduced in both these conditions. So this dramatic decrease in circulating T lymphocytes has been shown to be responsible for absence of cell-mediated immunity. So if you remember the last class where we were trying to understand the secondary response for the T cells, we have talked about the graft rejection. So there's a simple experiment performed where certain grafts can be uh, say implanted into some foreign strain, right? So your body is going to reject it because of the T cell mediated immunity. Now, in this condition with thymectomy or with in this disease condition, the, such rejections were not so robust, right? So, which show that the cell mediated immunity is really compromised in absence of thymus. And that's how people understood that, okay, it must be a site for maturation of your T lymphocytes because those are the primary mediators of cell mediated immunity, right? That is a discussion about the secondary lymphoid organ called the thymus. So these are very factual things. So we are trying to look at the important points relating to these things. If you must have also encountered these discussion during your recorded sessions with the faculties of the course. But if you have any doubts from there, that you can interrupt me in between and you can discuss this, right? Okay.
Excuse me,
So
ma'am.
yes,
Non-self antigens is presented by the APC.
non-self, yes.
So, ma'am, how they are reaching the thymus as they were not, let's suppose if they were not encountered by the body prior, so how the antigens are reaching to the thymus?
See, yeah, I understand your question. See, so this process of thymic selection, essentially during the process of thymic selection, there is say a purposeful subjection of the immature thymocytes to a lot of antigen, right? So what gets not what cells do not get selected for, that is in control of my body, right? So cells that can itself react to any antigen is not requiring the MHC molecules. Those cells are taken to death. The self antigens are there in my body. So you can have either of the antigens taken from different cells. Maybe every cell in the body also has a lot of receptors, a lot of markers on its surface. So maybe so it's a, how the body actually does it is not something that I know of, and I don't think it is even known that how exactly every antigen present in the body is presented to the developing thymocytes. But we do have the antigen reserve in our body as we are developing. So this is something that is also happening primarily during the embryonic stages. Okay. So you have to remember that even your maternal circulation, the blood from there, everything, it's not completely so the blood mixing do not happen, but we do get some antigen because we are already immune to certain infection while we are newly born as well, right? We also have the role of certain immunoglobulins crossing the placenta and all, which is also helping in that part. But coming back to this point, the self antigens are already being presented while the embryo is forming to the thymocytes inside the embryo's thymus. And whatever, if they react with if the cells are reactive, they are taken for death. So the apart from that, if we have any other cell which is non-reactive to self, those are the ones that can react to maybe anything that is not reacting to say uh, maybe a white color antigen. If I think of it like just two cases, a binary situation, if it is my own body and it is reacting, it is dying. Anything that is not my own body, those cells are selected for. So those are the cells that we say can bind the foreign antigens and are reaching the secondary lymphoid organ. So anything that is not mine, we say that these cells will react to foreign antigens. That is the summary. So it is not possible exactly, because for a neonat to actually have all the antigen present in the environment coming in, right? There's also certain sort of, so these sort of the thymic development, the thymocyte maturation, they do not even stop after birth. So while so this is a condition of before birth. After birth, while we are also having a lot of vaccination and such things happening, there how the foreign antigen interaction is happening is clear. Apart from that, if while we are just about in the environment, the thymocytes are getting matured, we are gradually, the newborns are actually the newly borns, the infants, the young adults, though they are always encountering a lot of antigens in the environment, right? So they can also get be presented gradually to the developing, the maturing thymocyte. So thymocyte maturation is not something that stops before we come out. It starts there, But as we are encountering all the foreign antigens, the process continues, and that's how and reacting to foreign antigen, those cells are getting selected for. Does it
Okay,
answer your
ma'am.
question?
Yes, ma'am, up to some extent it has helped me. And ma'am, in this stage two, we call this as naive T cells or not?
We'll come to it. No. Right now, they are just immunocompetent, mature thymocytes. The
Okay,
mature
ma'am.
thymocytes actually then differentiate into the naive cells. The naive cells means they have certain other molecules present on it. So the naive cells can then interact with the antigen. So you have one precursor to naive and mature cell. So it's at two ends of a bifurcated road. We'll come to it.
Okay, ma'am. And ma'am,
Mhm. Go ahead, I can hear you.
ma'am, if the question is somewhat like this, that T cells come across in contact with the antigen, where? So the answer should be thymus, or it must be a part of a peripheral
No,
lymphoid organ or
peripheral
not?
lymphoid organ. Right now, the stromal cells, they are just purposefully subjecting the thymocytes to different antigens. It is not the actual antigen that the thymocyte has to react. The question is that in which organ antigen encounter is happening. So for that, the first thing that should be is the actual path is the actual problem, the actual antigen which has come into the body, which now has to be taken care of. That do not reach the bone marrow or the thymus. You only have see. Okay, it's a very good question to overall visualize everything. We have antigen presenting cells everywhere in our system. It's entirely there in your blood circulation and everywhere from where it can migrate. So these APCs can migrate with the processed antigen, can migrate into the thymus or inside of the bone marrow, they can be taken in. I'm not sure about the bone marrow, but inside the thymus, so these cells, they have not brought in the antigen which is inside the thymus. From somewhere else, they acquired this antigen and the cell has now come into the thymus. So only for the purpose of presentation. But in the system, in the secondary lymphoid organs, you have the antigen directly coming in from the different body fluids. You have the antigen directly coming in. There, these antigen presenting cells are processing it for the purpose of presenting it to the T cells or the B cells that you give a response. Here, the purpose is thymic selection. In the secondary lymphoid organs, the purpose is to have an immune response against the antigen. So there the antigen is also present, there the APCs are also present, and we also have the lymphocytes present. So it's a war field, it's a battlefield. Here, it's a practice ground. If so, that sort of analogy can help you understand it.
Okay, ma'am, I got that point. And ma'am, this immunocompetent does not have any role to play with the thymus size as it decreases as the person becomes aged.
See, as the bulk of the thymus is decreasing, so if the organ bulk is decreasing, definitely the cells that is present inside it, the processes of selection and everything that is happening inside thymus, all those things are going to get compromised. So cell number will go down. This selection process is going to go down. That's why in older age, a lot of autoimmune disorders can come into the picture. That all happens because of. So this is one part that can be responsible for those disorders. But that's not the only thing that is responsible for it.
Okay, ma'am. So, ma'am, it can be said that it can be a reason that why old people are getting affected to the outside environment very faster.
Yes. Yes, yes, indeed.
Okay, thank you, ma'am.
Okay, so with this, the next primary lymphoid organ, which is pretty straightforward. So it is a site for the bone marrow is the site for B cell and T cell origin, but also the site for development of B cells. Again, we have something called as the stromal cells beside the bone marrow, which is going to present the antigen to developing B cells and make sure that before they leave the bone marrow, they are immunocompetent. Similar to there over MHC selection and thymic selection was going about. In bone marrow also very similarly, there is a selection for B cell with so that only the cells which have an antibody that cannot react to self antigen is allowed to get into the circulation. So it's exactly the same concept, just that over here, the context is in terms of antibodies because those are the B cell receptors as well and B cells. Everything, the concept is same. Now, before we get into our secondary lymphoid organ, which is of which lymph node is a prime structure, we'll just have a quick summary of what the lymphatic system actually, how is it coming into place and what is the exact function of it, right? So in the lymphatic system, what is happening? So how is a lymph forming, right? We have these capillaries, the blood capillaries, with, wherein we have the heart pumping the blood at quite some pressure. So whenever there is a fluid flowing through a tube which has some sort of perforations in a very high pressure, it is possible that some some liquid part of it is going to seep out of the vessels, right? So blood also has a lot of cells, right? But the cells is not going to seep out. So what is seeping out is just the fluid part of the blood, which we called as the plasma, right? So this plasma, first it is going to be there in this narrow space between the blood vessels and the lymphatic vessel, which is called as the interstitial space. Since the fluid is actually coming in the interstitial space, we say that the fluid is now named as interstitial fluid. The body ensures that most of the plasma that is seeping out of the blood vessel is taken back. So, whatever is the fluid coming out, most of it just actually gets back, right? Through the play of pressures, this is again going to go back because anything that is flowing at high pressure, there is a chance that this is not going to allow it. We have certain other mechanism which ensures that this plasma mostly is taken back. But whatever is still left, there has to be no loss of anything that is produced in the body. That fluid gets inside the lymphatic vessel and is called as lymph. So lymph is nothing, it's the seeped out plasma component of the blood, which is now flowing through the lymphatic vessels. It is important for you to understand that we say this a lot of time that there is edema, there is swelling in the lymph node, or there is accumulation of body fluid in case of some infections, or if you take certain medicines, we say कि हम कहते हैं पानी भर गया है. So what is this pani? This is nothing but the lymph that is there. So what can really help such situations? If you have heard, the doctors would often recommend that you say if you have fluid accumulation in arm, they ask you to move it. Or if there's fluid accumulation in your legs, they ask you to do some walking. So the pumping of your body muscles, that is what keeps your lymph flowing through. The blood is being pumped through heart, right? But over here, the lymph flow is ensured by usual contractions of your body muscles. So this lymph that has actually now formed, it flows through a separate set of arteries and veins and venules called as the lymphatic vessels. So just some terminologies. Okay, so this lymphatic vessel, the lymph node, the different associated structures, they all together form the lymphatic system. Now, coming to it from the point of view of immunology, So there might be a foreign antigen. While the fluid has seeped out, this foreign antigen has also come along with this fluid and it is now trapped inside the lymph. So this lymph that is going to reach, so lymph also drains all the interstitial tissue spaces, the gaps between. So these lymph capillaries, if you see, they form a very elaborate network. So this lymph is reaching all your, it's draining the tissue spaces to bring about whatever are the fluids. So if you see that this is from where the lymph started to flow inside the lymphatic capillaries, it is reaching the lymphatic vessels. From lymphatic vessels, it can now reach the lymphoid follicles, or it is also entering what we are going to see, the lymph node, right? So just a fact that the lymphatic vessel, the largest lymphatic vessel is the thoracic duct, which is going to return this lymph to the subclavian vein. So vein that is going carrying blood. So whatever is the lymph present inside the thoracic duct, which is the largest lymphatic vessel, this fluid is ultimately returned to the subclavian vein, which is taking it to heart. So that is how the fluid volume of the body is ultimately maintained. So your excess and nothing goes to waste. Your heart is again going to pump it back. Again, there might be some loss, but ultimately it is again going to get recovered from the body. So inside these secondary lymphoid organ, which is the lymph node, the antigen lymphocyte interaction occurs, which transforms the lymphocyte, the mature lymphocytes, into a activated state, activated lymphocytes. Okay, how is this happening? So, what is the organization? So, do not be afraid by the mess. The diagram is just this simple, where you have the lymphoid organ. This is the entire, so again, the cross section of your uh, lymph node. If you see the cross section, there is something called as cortex again, medulla, the inside part. There is also a diffuse zone which can be demarcated as differentiating between the cortex and medulla region, which is here named as paracortex. So you have cortex, paracortex, and medulla. Here we will study about three niche. There we had the medulla and cortex. We saw immature and mature lymphocytes present in either of it. Here we have three zones: cortex, paracortex, and medulla. So if we see the cortex here, so there they have just shown the it's a less elaborate structure where the drainage of lymphatic vessels have not been shown. But here they have also shown how the lymphatic vessels are draining inside the lymph node. So if you follow or if you trace carefully, we have afferent lymphatic vessel here. So afferent meaning the lymph is entering inside the lymph node. As the lymph is entering inside the lymph node, you have these projections from the, so again, we have the capsule. The capsule has, it has spaces for your lymphatic vessels to completely intersperse this particular node. So we have the lymphatic vessels that is carrying the lymph through entire structure of the lymph node, right? So from everywhere we have the lymph coming in. With the lymph, what also comes in is the antigen that is present in it. So once we have the antigen coming along with it, first it is going to be there. It is going to reach inside the cortex region. So the outermost region. So this until this part, it is the cortex region, right? So this is just a reference. It is very uh, difficult to visualize. I can understand in the first go, but just imagine the exterior most part, which is having your primary lymphoid follicle. So your cortex region has a lot of primary follicle. Primary follicles are these things. If you see carefully, this is some unorganized mass of cell present, which is being called as primary lymphoid follicle. Inside this primary lymphoid follicle, we have a lot of B cell, macrophages, and follicular dendritic cells. So dendritic cells, based on their morphological features, they can actually be um, classified into different types. So interdigiting means that it has elaborate processes that is completely. So interdigiting is you have digits and completely entering through every cracks and crevices. So if a follicular cell has that many elaborate processes, it is called interdigiting. Similarly, follicular dendritic cell, something which is comparatively more rounder. So follicular dendritic cells, B cells, they all together are just lying there. Okay, they are just waiting for some antigen to come in. As soon as there occurs an antigen encounter, your T cells, which is present in your paracortex region, along with the interdigiting dendritic cells. So you have T cells here, somewhere in the more middle part of, because we have the cortex, which is outermost, the paracortex, which is next, which is the zone for T cells. So T cells, which are present in your paracortex region, as well as the B cells in your primary follicle, inside your primary follicle, they begin to quickly proliferate, because proliferation is something that we are discussing from the very first class, right? So we have discussed proliferation in context of clonal selection, while the maturation happening, when there is selection happening. Prior to selection also, there's a lot of proliferation happening. So immune cell really invests in a lot of its energy in cell division. So as soon as this antigen encounter is happening, you have a lot of T and B cell proliferation happening inside of your primary follicles and in the margins of your paracortex. Gradually, with why is this proliferation happening? So with every subsequent class, we'll try to bring things in the same picture, right? You all have to join again. Okay, can I expect you all to please join back again? And let's hope that's the last joining and we'll be able to sum up our class in the next 40 minutes. Or you, okay, so if you recall, when we talked about the endothelial lining, so arteries are thick walled, but you should know that along with the arteries, we have them branching out into capillaries, right? So the capillaries have very thin walls. You can you if you have some
Yeah.
background of your so from the thin walls of your capillaries, il- assisted by this high pressure of blood flow, the cells can seep out. Sorry, the plasma can seep out. Am I-
Okay, ma'am. And ma'am, edema and interstitial fluids means the same?
Edema and interstitial fluid. So no, edema is the case where there is a swelling. But this interstitial fluid is what the swelling has occurred from. So when there is fluid accumulation, there is a lot of swelling. Swelling is called as edema. Why is this edema? Why this edema might be happening is because of accumulation of interstitial fluids.
Ma'am, the main reason for the swelling is this accumulation of interstitial fluid only, or it may be different.
It's not exclusive, but prime see your swelling can also be because of some cuts or breaches on the skin and something like that, right? But we read about there is tumor, calor, dollar. So there we were seeing that the tumor is coming because of a lot of cells and everything coming in. So the pus and everything, the debris is accumulating. So any sort of accumulation, any sort of accumulation is going to give you swelling, but fluids are primarily responsible for the swelling that we often encounter in our bodies. So which fluid? It's primarily the interstitial fluid, but nothing in biology is exclusive. And so that's what we also learn in research. Even if you find and feel like there's something which is exclusive, we never comment on it stating that this is the only reason, because you always have possibility of newer discoveries and something else might come up. So primary reason for edema is definitely the accumulation of interstitial fluid. but there might be a chance of multiple other ways by which your fluid accumulation, by which your swelling can result, by which edema can form.
Okay, ma'am. And ma'am, what is the role of the lymphoid follicle that is mentioned in the diagram that you have shown prior?
We'll just resume from there. I am not able to do this upgrade. It is taking a while. Maybe I'll just switch back to the class. Okay. Okay. So primary lymphoid follicle, essentially, it's a group of cell that to just bring you back. It's just a group of cell which is some having some sort of roundish organization. So it's arranged in a follicular manner. When there is an antigen that is, so let's just say that this is some antigen coming in. It is interacting with whatever cells are present in your primary follicle. So your macrophages and follicular dendritic cells, when they are encountering the antigen, they are going to have your MHC molecule, these are antigen presenting cells, and they are going to signal that something has come in and we have to counter this. So what do? They quickly proliferate. Why is this proliferation happening? Because there has to be generation of diversity, right? So we have the mature, so this proliferation that is actually happening is, so we have clonal selection occurring. So whichever anti cell with a particular receptor is able to bind this antigen, they quickly proliferate. Now they are called, if you remember from clonal selection, these are now selected for. So we have selection of lymphocyte happening in presence of antigen, and they now organize into a secondary follicle structure. Okay, so we have a lot of cell proliferation happening in response to presence of this antigen. Who presented this antigen are the antigen presenting cells in your primary follicle. In response to that, the proliferation happens, the antigenic selection happens, and the activated lymphocytes are now organized as a secondary follicle. So the structure of secondary follicle is such that we have some germinal centers, right? If you see that we have some round outer layerings, inside of which there are some cells. This uh, this structure is known as the secondary follicle, and the central of it is the germinal center. What is exactly present in this germinal center is the B and T cells, which are still interacting with the antigens, which is being presented to them by the follicular dendritic cells, the B cells, or the T helper cells. Once this interaction has happened, the selections have happened. What happens is your plasma cells. So if you remember, so T cells essentially have to take up the function of killing. So inside this system, we primarily, because the antigen has come in, right? Cytotoxic T cells come into the picture when they have to kill a cell. Here, if we are saying, so if you see, we have the T helper cells, because the role of T helper cells is to release cytokines, and so their interaction with the antigen here is going to release cytokines which can reach back into the bloodstream. The B cells, which now differentiate as plasma cells, they also need to reach the bloodstream. So whatever has to reach the bloodstream, that moves inwardly more towards the medulla region. So anything that you see more inwardly, so these cells maybe that you see here, they are all activated lymphocytes which are capable of giving a effector response. So these cells are then ultimately moving out from the medulla, from the efferent lymphatic vessels. And people have actually tried to quantify the number of lymphocytes in your afferent lymphatic vessels and efferent. So afferent is coming in, efferent is leaving. So efferent lymphatic vessels have 50 times higher concentration of lymphocytes. This, so as we were talking about proliferation, uh, it's obvious to expect that the increase in number is because of lymphocyte proliferation that has happened inside the node. But this only constitutes 25% of this increase in number. Primarily because of the antigen encounter with the lymphocytes, we have many previously formed blood-borne lymphocytes that can now migrate into the node. So all these lymphoid organs are also having vascular supply, right? If you see here, it's a venule over here. So okay, so this line you see, it's the lining of a vein. So what do vein have? They have blood. Along with blood, they will also have all the cells of your immune system. Along with the RBCs shown here, we also have lymphocytes which can move out from these venules. And where will they come in? They are going to come inside these regions, right? Where they are now interacting with your antigen. So the primary increase in this number of lymphocytes in the efferent lymphatic vessel is because of all the blood-borne lymphocytes that have migrated into the node by passing through the endothelial cells lining the venules, which is supplying to the node. So this way of trapping the antigen is most important for your any antigen that is there in your local tissue. So now that you have understood how antigen is actually coming in your secondary lymphoid organ, let's also understand that where this lymphoid organ is placed. Uh, just a background in maybe physiology, the circulatory system. You would know that lymph nodes are actually present very nearby all your say all the important organs. You have lymph node almost placed at every nearby every joint. So you have it here in the armpits, maybe on the backs. So in very closed associations, you have the lymph nodes placed. Why has the body not made one node and have spread it across? So that every antigen in the localized tissue gets a very fast chance of getting inside the lymph node by chance, where we have our army station, which is ready to fight the antigen. As soon as the army identifies this invader, it quickly comes out, reaches the place where the antigenic challenge has occurred, and it is trying to eliminate that infection from there. Okay, so this lymph node is primarily the way by which your antigens from the local tissues is taken care of. So that is all that we had to understand about the lymph node. We have also discussed that this cortex is actually having B cells. So this is also known as thymus independent area, whereas the paracortex is known as thymus dependent because it is having T cells, and T cells have essentially come from thymus. So in the case of thymectomy that we were talking about, it is noted so that the paracortex is devoid of T cells. So that's why it is known as a thymus dependent area, and cortex is known as thymus independent area. The next secondary lymphoid organ is the spleen. So lymph was for the local tissue-borne antigens. The spleen is primarily the place for encountering blood-borne antigens, right? Which means the systemic infection. So system is something which is in your generalized body, in the general system that is present. So if we have, say, a bacteria which is freely circulating in the bloodstream, that sort of antigen is known as the antigens present on it is known as the si- uh, just is known that are the blood-borne antigens, and they are primarily get encountering the lymphocytes in the spleen. So similar to the lymph nodes, which was being drained by the lymphatic vessels, spleen, which is bringing blood-borne antigens, so it must be getting supplies from arteries and veins. So the splenic artery, so which is bringing fresh blood inside the spleen, They are the one which basically carries these blood-borne antigen along with it the lymphocytes, the mature immunocompetent lymphocytes, right? If you now look at the cross section of spleen, you will see that again the outer layer is having a capsule covering. This capsule is again having certain projections going inside the 3D bulk of the organ called the trabeculae. And we now have the arrangement as a red pulp and white pulp because of we have RBCs coming here, right? So whenever there is RBCs, there's going to be reddish, it's going to be having a reddish color. So we have a red pulp where we mostly have the RBCs, very few lymphocytes, and some macrophages. So this zone is known as the red pulp zone. So so spleen is also the graveyard of RBCs. So it is expected that there's going to be blood and a lot of dead RBCs because they have very short lifespan. And along with that, we have a few macrophages and lymphocytes. Apart from the RBCs, we can also expect the arteries to be bringing in the antigens, which is going to get a chance to interact with the lymphocytes stationed in your white pulp region. So similar to what we saw in the lymph node, the white pulp region also has these primary follicles and the secondary follicles which have a germinal center. Now, again, like the lymph nodes, primary follicles is something that is rich in B cells. We have a diffused zone between this red pulp and the white pulp, which is known as the marginal zone. This marginal zone also has a lot of lymphocyte, the macrophages, the antigen presenting cells in general, and we have closely associated periarterial lymphoid sheath. So what the name suggests, so you see this is the artery. The artery is draining like this. We have a lymphoid sheath associated with the artery. So if it is associated, we have periarteriolar. So surrounding the arteries, we have a lymphoid sheath. So that is how the name nomenclature has been done, also known as PALS. So this PALS is where a lot of T cells are stationed. Now, whenever this blood that is coming in, so it empties inside the spleen. So we have the antigen, which is first coming out in this marginal zone where they are trapped by the dendritic cells present in your marginal zone. Once the dendritic cells have trapped the antigen, they are going to perform their job of antigen presentation. And we are going to see a lot of similar way. We are going to see a lot of B and T cell proliferation, their activation, then the organization of primary follicles into secondary follicles, where again the germinal centers are going to have the activated T and B cells, and these cells can then move out from the spleen to take care of your antigen. So just the same part. So over here, as the blood is getting emptied in the marginal zone, the antigen is presented to the lymphocytes in your the T cells, and the activated B, because the antibody here is going to directly identify the antigen. So the presentation part is only done in the periarterial lymphoid sheath, the PALS. Now, this activated B and T cells are now going to move from the primary follicles, develop into the secondary follicles, the germinal centers, which have a lot of dividing B cells called the centroblast and the plasma cells, and eventually they can come out in the blood so that they reach the site of antigenic challenge. Now, the last secondary lymphoid tissue that we are going to discuss is the mucosa associated lymphoid tissue. Yeah. So the mucosa associated lymphoid tissue. So as the name suggests, mucosal linings, we have witnessed them line all our important tracts in the body. So the respiratory tract, the digestive tract, the urinogenital tract, all these tracts have the mucosal lining, and we have some lymphoid tissue associated with it. Now, if we take together the total surface area that is being covered by all these marks in general of all the different tracks, it's somewhere around 400 square meters. So it's a lot of surface area that is being covered by a lymphoid tissue. Now, these are the major site, because if you just imagine, before an antigen is going to reach inside the blood, how is it reaching there? It is reaching there through the lumens of your body, through your digestive tract, through your urinogenital tract. So those are the first openings from where a lot of antigens are getting inside, right? So these are the major site for the entry of your pathogens, and most of the uh, the most active area of antigen encounter are also these mucosa associated lymphoid tissues. So they can be a loose bunch of cells just lining certain some places in your entire tracts. So those can be very poorly organized. For example, we have some just some cell clusters. So far we're seeing that there's a capsule, there's some blood flow, there's organization of the organ. Inside the organ we have follicles, different. There's nothing like that, just some loose cells which are just packed there, which are just waiting for some antigens to come in. So that can be the case. Or we can have somewhat more organized structures, but just not as, it's not like an entire organ, just some organization, like the tonsils, appendix, or the Pierce, which is present in your intestinal submucosa. So, going one by one. So if we see, so this is your tonsils. So if you see here, we just have so there are three kinds of this. One can be your lingual tonsils, which are at the base of your tongue, the palatine tonsil at the back of your mouth, or we can have the roof of your nasopharynx, where the which we also call as the pharyngeal tonsil or the adenoids. So you might have also come across some people who have inflamed adenoids, and they might have want to like get, so because of the inflammation, it's also swollen, and they have some breathing difficulties. So adenoids can also be removed maybe, but yes, it is going to suspect you more to nasopharyngeal antigens, but it's not completely done. So just an example. Okay, so inside these tonsils, if you see, it's just some follicular arrangement present here. So these are the tonsils, just some follicles with some B and T cells. That's it. So far, we were looking at those elaborate organs and uh, different distinct anatomical features of it. But we just have some lymphoid tissue where there's some follicles. And as we were discussing, the follicles essentially have the B and T cells. And if there is some antigen that it encounters, it gets a chance to proliferate and take care of it. Right? So tonsils are nothing but these are small nodular structures which consist of a meshwork of reticular cells and fibers, which is interspersing all the lymphocytes, the macrophages, and the granulocytes. So just some cells to present it, and some lymphocytes associated to this antigen presenting cells. A little more, so it's somewhat different, is the mold of your GI tract, the gastrointestinal tract. In the gastrointestinal tract, so what happens is, so just imagine it like we have our food pipe. Okay, excuse me. So this is your digestive tract. There must be some lining, some cells lining your margins of the digestive tract. This is a lumen. It's a open space, right? Here we have the underlying vessel, blood vessels. So, and the blood vessels are also going to have some cells lining its lumen. So we have the vessels where the blood is flowing. We have the lumen, where the food is coming from. Imagine a case so this side of the system is the lumen, and here we have the blood vessel. Now, the lumen, along with the food, there's a case where an antigen has come in. For this antigen uh, to be taken care of by the effector cells, it has to either this has to cross into the blood so that the lymphocytes in the blood can take care of it, or we can have some in between battleground where the interaction of the antigen with the activated or the effector cells can happen. There is a cell called M cell, which is a antigen transporter. What it does, so like other normal cells of your intestinal lining, you might recall from digestive system that there's a lot of villi present on the cells lining your intestine. But this cell in between your cells with the villi, they do not have any sort of villi on it. They lack the villi, and they have some possible porosity through which the antigen is entering inside the cell. And this cell is also in continuity with the blood vessels. So from extravasation process, you can have some cells of the blood crossing the lining of capillaries, and they are going to reach the so your tissues are essentially compromised of cells, right? So these cells can actually move into the interstitial space between your M cells and other cells of the intestine. The antigen also has come inside. We have effector cells moving into it. And this is a temporary battleground where the antigenic challenge is met. So if you so this is just one. So this small part has been further say magnified in this portion, but this is how your entire digestive tract lining looks. So we have a mucosal epithelium where we have a lot of villi projections from the cells lining it. Just some cells called the M cells, if you see the structure, they have a small space in it. So they have a small space here, right? So this is where your antigenic challenge is taken care of. It is also possible that this lamina propria region have more organized lymphoid follicles, and once the antigen is crossing through the M cells, they reach this lamina propria region, and it is now met by these organized lymphoid follicles. The antigen challenge is there. They are going to release some effector cells or the plasma cells. These plasma cells can again come back into the lumen where they release the antibodies, right? So plasma cells are the antibody bound cells. These antibodies in either the cell itself can get trapped, and now it is called as the intraepithelial lymphocyte. It is going to be there like the memory cells, or it can secrete out its receptors into the lumen. The only antibody that can actually come into the lumen is IgA. So this comes out in the lumen and it is now going to interact with the antigen. So this is how your mucosa-associated lymphoid tissues are taking care of preventing any sort of damage caused by the antigen. Okay, so this was about the lymphoid organs. You all can take 2 minutes and just see if there's anything that you have to ask me about this. And then we move on to, so we have discussed this part very briefly, the part of maturation and activation of B and T cells. But now we will try to put things in context in terms of lymphoid organs. So at which place, what is happening in the lymphoid organs? Okay. So before I discuss for B cell and T cell maturation activation, I like to give you small background about antibody molecule, because then you will be able to better understand it. Just some structures. I am sure you have encountered it already in the recorded lectures, but for me to put things in the same continuity. So if you are aware already, this is how our antibody molecule looks. It essentially has two heavy chains and two light chains, right? And there's some disulfide bonds and everything present there. Along with, so when they are bound to the cell, they also have some transmembrane domains which keep them embedded inside in the cell membrane. Apart from that, if I talk about B,
Ma'am,
yes,
ma'am, this is the antibody that you have drawn on the cell.
you don't agree with the structure?
Yes, ma'am, I don't agree with the structure because I think
Yeah,
that the two light lines should come the outside and you have drawn in between. So,
you're right. They do come on the outside. I thought I would just escape with the gradual diagram. Okay, now this is okay.
yes, ma'am.
I am again being shown that this is going to end in 10 minutes. Okay, let's just quickly do this and then we'll join back. Okay, so if we have, so the idea is that yes, there is a membrane bound antibody molecule. Apart from that, there are also two other molecules present on the B cell membrane itself, which facilitates, so whatever is the antigen binding here, it binding of antigen essentially has to, so it's a signal to the cell, right? That something is bound, I have to do something. Even if it has to proliferate, binding of antigen, even if that has to trigger proliferation, how does the proliferation should, the signal for proliferation has to come from inside the cell. The antigen has bound a receptor on the cell. So for this signal to transduce into cell, the nucleus, the cytoplasm of the cell, we have two other molecules associated here. They are called alpha beta molecules. Okay. What is it? What do they do? There's still a couple of more classes before we get there. But you must just know that we have two other molecules, and this complex in its entirety is now called the BCR. So antibody is the receptor, but for its signal transduction into the cell, it needs two other molecules. Keep that as a background. Now, the B cell maturation actually happens in two phases. One is your antigen independent phase, and the other is your antigen dependent phase. And as the name suggests, one phase is going to require some antigen interaction, and some part of it is not going to need any. So, from a stem cell, so we'll start from the very beginning. So, there is a stem cell, the lymphoid progenitor. So from HSC, we will have the lymphoid progenitor cells. From there, we have the B cell progenitor, and ultimately we have a pro B cell. So this pro B cell, just excuse me, I'm just drawing this as an antibody molecule. So this has the Ig. Okay, I'll draw the antibody fully. This is Ig alpha and Ig, the alpha beta molecule that we were talking about, right? So pro B cell only has these two molecules being expressed on its surface. Now, we also talked that there is some sort of gene rearrangement that is happening, right? We were trying to link it with recombination. That is how the rearrangements are happening. But for now, just know that, so we have the heavy chain. So it is clear that we have uh, two different proteins placed together, right? So So we have something encoding the heavy chain, some other gene encoding the light chain, and together they constitute, because of the disulfide bond, they are now the same protein molecule. So two genes and one protein. So at any point, if something is not clear, please stop me in between. Okay. Okay. So when the Ig alpha and beta molecules are expressed, at this point there is a lot of heavy chain gene rearrangement. Okay, I will have to give you some background. So what happens if this is the entire gene that is going to code the heavy chain? There are some sites that allow the activity of restriction endonucleases, right? So the recombination that we have been saying so far, what happens is you can have, so restriction endonucleases are just enzymes. You can call them molecular scissors, right? We can have, say, RE1 coming in. It is going to cut at this place. This end can be joined here to make, say, one combination. So if this is segment 1, 2, 3, and 4, this is segment 1 and 4 that has formed. If, say, RE2 has been cut, we can, if both of them have cut, so we can have the segment 2. And if now this has to join back to 4, we can have 4. So this sort of rearrangement is happening, right? So heavy chain gene rearrangement, it's just some cut and paste that is happening. For now, that is what you should visualize it as. This cut and paste is happening at the stage where pro B cell is becoming a pre B cell.
Excuse me, ma'am.
Yes, go
This
ahead.
gene rearrangement is taking place inside the nucleus of pro B cell.
Yes, the genes are present there. But ultimately, the expression is going to be on the cell surface in terms of the antibody molecule that is going to now become expressed on the surface of your pre-B cell. So we have the pre-B cell, which, why am I calling it a pre-B cell? Because now also the antibody molecule that has formed here is not what you find on your plasma cells or say the activated B cells, right? So this is some sort of precursor chain that is there. Okay. Now this pre-B cell is again going to undergo, because only the heavy chain arrangement has happened. Now this is going to undergo light chain rearrangement. Once the light chain has also rearranged, this is known as, okay, it's still in a immature B cell. But now we have the properly formed cell-bound antibody molecule in place. The first antibody molecule, that is, definitely we still have the Ig alpha beta. Okay. The first antibody molecule that is expressed on the surface of the B cells is the immunoglobulin M. So membrane bound, that's why we put a M, and immunoglobulin M. When we read about the different types of antibodies, which is just in our next class, there are 5 types of antibodies. IgM has the highest affinity, highest avidity of binding the antigen. Now, what do I mean by that? So, if you look at this molecule, there are 2 possible antigen binding sites. One is this site and one is this site. Your IgM exists as 5 antibody molecules bound together using something known as the J chains. So as if 5 molecules are attached together, right? We'll have one here, one here. Now, since it has 5 molecules, it exists as a pentamer And the first go, how many antibodies it can bind is 5. When it is bound to the cell receptor, it is a monomer. But your B cells, when they are producing the antibody, what it is doing, the cell-bound receptor only is getting, it's just going to lose its transmembrane domains and it is going to get secreted. Once this receptor is secreted out, you have multiple B cells having these IgM molecules. Or maybe the same B cell can also have multiple IgM molecules. When they are secreted out, they are going to exist as a complex of 5 molecules together, pentamer. Now, this 5 molecules, when it is going to find the antigen, it's not like there is just one by one, there's just 1 or 2 particles of antigens in the surroundings, right? It's always in a huge number. So the body's challenge is to produce effector molecules in an even larger number. So when they have this ability of forming a pentamer, and that is why the body is smart enough to know that the first receptor that I have to make on a B cell, on the immature B cell, should be of the Ig5 make, IgM make, because when it is released, secreted, it can exist as a pentamer and bind multiple antigens together at the same time. Less than 1 minute left, girls. Just now with this immature B cell, the next receptor molecule that now comes into effect is so there is another Ig molecule that has to be expressed on its surface. It is the IgD molecule. So we already have the IgM, right? The alpha beta molecules. Now this make of the cell is finally called as the mature B cell. ठीक है? So far, while we are talking about proliferation, what is essentially happening in this proliferation is this. So you have a stem cell, the pro B cell coming, the pre B cell coming, the gene rearrangements happening, immature B cell, the Ig molecules switching. So all those things are ultimately forming a mature B cell. This mature B cell, so far, this phase is known as the antigen independent phase, because if you see, it has not required any antigen's presence. This is your antigen independent phase. This is antigen independent, and where is it happening? It is happening inside your bone marrow. So we were saying that the site of maturation is also development. So definitely the site of maturation is also bone marrow for B cell. Let's just remove this from picture. We have to take it in the following class. This is followed by this maturing B cell to enter your peripheral tissue, the peripheral lymphoid organ, and this begins your antigen dependent phase. Now we need an antigen, and in itself, your B cells do. So your B cells can respond, can identify the antigen by its antibody bound on its surface. So we have an antigen. We have 2 antibody molecules stationed on its surface, the alpha and B make, the alpha and beta immunoglobulins as well, and there comes in an antigen. Now, something was there which has to stimulate this B cell. If you remember, it was from the cytokines. So the cytokines can actually come in from, so we need the cytokines coming in. The cytokines can come in from either the T helper cells, or we have the macrophages and the dendritic cells that can bring about the cytokines, that can release the cytokines. So in presence of this cytokines, in presence of this antigen, it switches to a So this was a mature cell. It switches to a activated B cell. This activation process, this is where, so no feature changes here. This activated B cell actually requires this, the process of this activation requires the alpha and beta immunoglobulins. Because as I was saying, they are the transducers of whatever has happened. The antigen has now come in. The signal has to be transduced into the central of the cell. And they have to undergo a lot of clonal expansion. So the mature cell here, because so once you have your gene rearrangements happening here, the antigen commitment is happening. Why so? Because whatever is the combination of rearrangement that has formed, that is leading to the cell's antigenic commitment. That this is the antigen that I can respond to. Once you
Ma'am,
have,
will you please repeat it again?
when the rearrangement has happened, so essentially the diversity that we were achieving, it is coming from the rearrangement process. But once the rearrangement has happened already, we have a certain combination that has already formed. With this combination now determines the antigenic specificity of your B cell. So antigen commitment has happened at this stage. Is this clear? That it can only respond
Not
to a
clear with
particular
the
type
last
of.
statement that you have said.
Sorry?
That from the antigenic specificity, it is not clear to me.
I am okay. So antigenic specificity is coming from the rearrangement that is happening. Why so? Because as I was saying, it's a lot of cut and paste, the 2 coming to 4, the 4 coming to 3. All this sort of rearrangement, once a particular uh, fixed arrangement has formed. So imagine it like maybe it's like a clay, say. So you have a clay, you can mold it into any shape and structure. But once we have arranged this clay and we have allowed it to say dry in the sun, it is now fixed. Now you cannot mold it. So whatever is the shape of the cavity that has been formed, if you just imagine it like that, whatever is the structure that has formed at the cavity, it is now fixed. So only the antigen that can actually fit inside this cavity can be bound by this particular B cell. Any other structural features which cannot fit this cavity is not going to be bound by the B cell. So binding is essentially required for the B cell to recognize an antigen, right? So antigenic commitment has been defined here because the structure, the clay has already hardened. Now you cannot mold it further. So there is antigenic commitment that has happened. Is this
Okay,
okay?
ma'am. So, ma'am, it is happening in the antigen independent phase after the light gene
No.
rearrangement has been taken
See,
place.
antigen has not been bound yet. We do not have the antigen coming in yet. But the rearrangements have occurred that is going to determine what kind of antigen it can bind.
Yes, ma'am, that's only. So at that point, we are calling that as antigenic commitment because from there, no further changes can be made on the antibodies gene, right?
Yes, right. It's not like if a different make antigen is coming, the rearrangement is going to change in response to the antigen. No, that is not going to happen. So antigen has
Okay.
not come, but the specificity for any sort of antigen has been determined already by this rearrangement happening. So the rearrangements that are actually happening is random. The body doesn't know that this rearrangement is going to be able to bind to this A antigen or B antigen. It is producing a lot of random rearrangement so that every possible combination has been formed, right? So we were, if you remember, it's 10 to the power 10 specificities that can be formed by these rearrangements. So 10 to the power 10 types of antigen can be recognized by the B cells alone. ठीक है? So antigenic
Yes, ma'am, but
commitment has.
they will be recognized by the different antibodies structure that are present on it, right?
Hmm. So B cell receptor is nothing but the antibody, right? So we have different types of antibody in terms of the typing is actually happening based on the features. But there can, every antibody has a, so this is going to be more clear in the next class itself. There is something called as the constant region and something called as the variable region. Based on the constant region, we have 5 types of antibody. But each type of antibody, the variable region is where the rearrangement part is mainly producing an effect. The variable region for each type of antibody can be very, very, very different. So you can have each type of antibody binding different types of antigen based on whatever are the amino acid sequence. So the amino acid sequence are actually defining what kind of antigen it can bind. Based on the rearrangement, the amino acid sequence of the antibody variable region is changing, and that is how the antigenic commitment is specified.
Variable region will not be changed further after it
No,
encounters the antigen, right?
no,
Because at
no.
that point, antigenic commitment has already taken place.
Yes. Yes. So the variable region doesn't change. Whatever is the specificity of variable region, whichever antigen is now being recognized by this particular cell is going to further lead to an immune response. This B cell has some sort of, say, specificity that, what happens? That doesn't, एक second, that doesn't meet the antigen's structural make, it is not going to recognize it. Okay.
Actually, your voice got break in start of the statement. What you have continued at the end till the end.
Yeah, ma'am,
So,
the IgD which you drew there, that's
mhm.
something which I didn't get.
See, it's just another, so there has to, for a B cell to be called as a mature B cell. So, as long as it was just having IgM, B and beta and beta and alpha immunoglobulin, it is called as an immature B cell. For this immature B cell to become a mature B cell, it also has to express a IgD molecule. Why is this happening is what we will talk about when we were discussing the next class of antibody. That what are the different types, what is the function, why this sort of switching has to happen. Okay. So that is to answer Mansi. Is that clear?
Yeah. So basically, it's like IgM has been formed, and after IgM, whichever class forms, so it's like you showed that IgD is formed there.
IgD has to form, not whichever class.
Okay.
IgD has to form. Okay. Membrane. So basically, there are just, so of the five
Two membrane
types of
bodies.
antibodies, there are
Uh-huh.
just two types which can have a transmembrane domain. It's IgM and IgD.
And IgD.
Right. So IgM,
Uh-huh.
after IgM, it is immature B cell. Once IgD has also come, it is a mature B cell.
Okay.
Right. And for
Okay.
Shamika, the variable region is actually already determined before the B cell is coming out from the bone marrow. The specificity of the variable region is defined. Now, whatever is based on whatever is the antigen coming in, particular B cell is going to be selected. How? The antigen binding to a B cell which is compatible of binding this antigen based on the variable region's specificity, right? It is going to bind that antigen. So antigen commitment has happened, but no antigenic encounter is happening in the bone marrow.
Okay, ma'am. So, ma'am, I can say that the all the B cells that are forming in the bone
Mhm.
marrow, having IgM and IgD, are not having same variable region. The variable region can be
Yes,
different, but the
yes,
antibody will be same, right?
yes,
Uh, IgM and
yes,
IgD will be only there, but the variable region can have different sequence of amino acids
exactly,
and that can bind to the different types of antigen that they encounter.
exactly, exactly. Perfect. ठीक है?
Okay, ma'am. And ma'am, IgD antibody formation does not require any gene rearrangement, or it has
It does.
already
It
taken
does.
place.
No, it has
It has.
it goes through. So before an antibody molecule is expressed on the surface, it has also similarly here it has to be over here. It has already undergone all the gene rearrangement that is possible that happened before the IgM molecule came into existence. ठीक है? So gene rearrangement happens for every antibody molecule, every T cell receptor molecule. But it's still I think couple of more classes, I think 5 or 6 of them before we discuss that topic, because it's a Nobel Prize winning concept, so it's not very easy. I also really need to go through it again and again before I get comfortable in delivering the topic. Okay, we'll come here, right? This is okay. So for activated B cell, how is it getting activated? Based on whatever is the specificity of the antigenic commitment, we have the antigen encounter here. This antigen encounter is activating the B cell. How is this signal for activation reaching the cell nucleus, say? It is through the beta and alpha molecule. So these are basically signal transducers, and it has some, I think it's some tyrosine kinase signaling that happens, which is transferring the signal to the inside. And the activated B cell's role is to first proliferate, because it has to undergo clonal expansion. So this is some standalone concept that we have learned about in our first class itself, that there is clonal expansion happening, and then there is clonal selection. So what happens is activated B cells rapidly divides, and divides into what? So clonal expansion is cell divides into, you can call it as a duplicate. So duplicate is having the same antigen specificity. So clonal expansion is happening where this particular mature B cell that could bind the antigen, whatever is the combination of variable regions here, that is going to get selected for in presence of this antigen, and there is going to be clonal expansion, and this is the state of clonal selection. So clonal expansion and clonal selection is essentially the same thing. So because of clonal selection, clonal expansion is happening. Now finally we have its differentiation further into the plasma cells and memory cells. So your and specificity is actually determined here. Nothing changes beyond this point. Whatever cell is going to be able to, its respective specificity, it is going to bind this antigen. It is now activated. It is going to make exact duplicate copies with the same specificity, and it is then going to differentiate into either a memory B cell, again, which is having the exact same specificity, or a plasma cell. So in this process of differentiation, if I just talk about the plasma cell lineage, here the concept of class switching is going to fit in. So that is something we'll read in the next class, that what is class switching, why is that happening. But just a brief, that exactly how I was saying, that every antibody molecule, every type of antibody molecule has different features to it, and to best encounter these state of antigenic challenge, we have the class switching phenomena that comes into play. Okay, I think so do you guys have doubts, or else we will just further go ahead with 2, 3 further concepts? Or if you have doubts, we'll stop here and we'll take the questions first.
I'm having some questions.
Yeah, go ahead. Then we'll after the queries, and we'll end the session then. Hmm. Bolo, bolo.
Ma'am, you have told that at the end the clonal selection is taking place first and then clonal expansion will take place, or it is just the reverse?
It's clonal selection first, and clonal expansion is a consequence of clonal selection.
Clonal selection will take place first and then clonal expansion will
Hmm,
takes place, right?
hmm, hmm,
Uh,
hmm.
so the sequence in which you have wrote, is that correct?
See, I should have first talked about clonal selection, but it essentially means almost the same thing. But if this is easier for you to understand, we can move this. I can move it around, not a problem.
No, ma'am, it's okay. I can
Mhm.
understand that. And ma'am, some antibodies are already present in blood. So how they are formed? They are already formed from the mature B cells without any antigen encounter.
Sorry, I didn't. Can you just repeat it? We already have some antibodies in blood.
Yes, ma'am.
Yes, so any antibody to form, it is important. How did that first antibody come from? So we have some antibodies that actually come from the mother in the breast milk. But whatever is the antibody that has been formed by our own, so they must have come because of some prior interaction with some antigen. It's
Ma'am,
not
they
like
are
you have
formed.
a. Where are they
So they
formed?
are, no, ma'am, they are formed from the B cells. So they are formed from the mature B cells or from the activated B cells are only from that time they
Only
are
the
present.
activated B cells are going to differentiate into plasma cells, and antibody only comes from plasma cells.
Okay, ma'am.
Mhm.
Ma'am, T cells are below the primary follicles, so it is because of the fact that they required some antigen presenting cells or the receptors that must represent the antigen on the cell surface so that the T cell can further can come in contact with the antigen and then differentiate.
Below the primary follicles, as in?
Yes, ma'am. In the diagram that you have shown, every time they are coming below the primary follicle. So I thought that.
No, no, that's not the case. It's nothing like. Uh, so see, I think okay, you're talking about this correlation that this is on the top and this is on the bottom.
Yes, ma'am. Ma'am, because B cells antibody is only the receptor that is present, so it not require any antigen presentation, while the T
Uh,
cells require, so they are taking the help of the lymphocyte that is present in the the APC cells that are presenting in the marginal region, I guess.
See, so the case is what you have to, if you look here, okay, this is also a secondary follicle, and this is a primary follicle without a germinal center. It has got, so you have to, instead of, so it's a transection, I can understand that, where you see the top and bottom, but essentially it is the out. So you have to imagine it like a 3D space. Maybe this can help. So this is a 3D. So let's say this is the spleen, right? Here on we have the arteries and the blood supplies coming in. This outer surface is the red zone, the red pulp, and nearby this artery, this somewhere in between the volume is your white. So this centric volume of white pulp, whatever is the outer space, right? So if there is say uh, another tube here, the outer space nearby this tube is the place where your primary follicles are stationed. So gradually the things that have to come back into the blood are towards the center of your hollow. ठीक है? यहाँ, if this is the outer surface, we have the primary follicle somewhere here, which is like uh, the outside of your white pulp, and the more centric region of your white pulp is going to have the secondary follicle. The reason for that is the the middle region is where your arteries and supplies were there, right? Now your follicle with your effector B and T cells, they have to ultimately come back in blood, or they have to ultimately come back in efferent lymphatic vessels. So if you look in the spleen, and if you also look towards this region, this is where it's closest for them to get back into the duct, into the lymphatic vessels. That's why they are close by the central region of the organ. It's not like top and bottom, it's more like more inward in the organ, because we have the vasculatures there and the effector cells have to come out of it. So antigen is drained from the outer surface, if you say of the lymph node. And if you talk about spleen, we have the blood here. So the antigen draining is also somewhere near the margins of the marginal zones, say. But then the mature follicles, the secondary follicles, are somewhere more central so that they can be taken back.
Okay, ma'am. And ma'am, in the secondary follicle only proliferation is happening in both the follicle, we can say.
There is proliferation that is happening in the primary follicle, and then they become the secondary follicle. So it's not like this primary follicle migrated out and have now become a secondary follicle or something. Your primary follicles are going to mature as they are moving towards the medullary regions into, so they're maturing into the secondary follicles. It is becoming more organized. Instead of lying around like a mass of cell, it has now undergone a lot of proliferation. It has got a germinal center. While this process is happening, it is also kind of, the follicle in its entirety is also moving more towards the central, the medullary region of the organ. So it's not like this has become this. It's like every this is going to become this. And this is going to move out into the blood.
From the germinal center, B and T cells will migrate into the blood vessels, right?
Yes, yes.
Okay, ma'am. And ma'am, the M cells lack villi extravasation. M cells lack the villi, and we can say that from villi to antigen can also came inside the
No,
body.
if from villi, the antigen could be, see, every surface, so every margin of the cell, so surfaces are constituted essentially by a cellular layer, and every cell might not allow something to enter it. So M cells are specialized cells which are able to transport the antigen. If the cells with a villi, the normal lining cells of the intestine could have allowed this antigen's entry into it, we would not require a M cell. And as I was saying, that if you see, you have the nucleus, the morphology of the M cell is actually allowing the antigen to come in, so that this space is utilized for the encounter. So this is not like it's a
Okay,
hollow
ma'am.
space, right? It's an extracellular matrix space. So your blood supply is bringing cells in the matrix space. You also have the antigen coming inside the matrix space, and that's how uh, this becomes your battlefield.
Organized lymphoid follicle, which is shown in the diagram, is having B cells only, or the T cells are also present in it?
It's both. I think so. Let's just read here. It's both the cells present, right? Of which many are CD8 positive T cells, but they are this particular. So I was telling there's a gamma del cell. So the T cells that are mostly present over here are of the gamma del uh, cell type. So we have cells with the gamma del type of TCR, but
Ma'am,
it's
we have
primary.
shown that TC cell and TH cell are differentiated on the basis of the CD8 plus and CD4 plus.
See, that is
So
not
this
the
is the
complete.
different. So
Yes,
this
this
is
is,
the
yes. So we have just looked into two broad categories of it. You must have also encountered, see, I think you only asked the regulatory T cells, the
yes,
uh,
ma'am. That question I have
follicular
only
T
asked.
cells. right?
Yes, ma'am.
So that's like not the complete picture. So basically, so these are cells which have TCR, exactly the same structure of TCR. It's just the foldings and the say the sequence of amino acids and things are varying based on. So if the sequence of amino acid vary, it is going to assume a sort of different folding, right? So this different folding is called the gamma and del instead of the usual alpha beta. So this is not exactly so the function of these cells are not known. So these are not doing killing function. These are not producing any sort of cytokines for helping some other cells. The function of it is not known, but it is there. So we have some CD8 positive. CD I was saying, so these are some additional molecules which is of CD8 type. But the TCR present here is gamma and del type. So this gamma and del type of T cell with a CD8 plus molecule is not performing any known function. Essentially the major effector that is coming in picture is the plasma cells.
Okay, ma'am. And plasma cells are formed from the B cells differentiation, right? Right now we have shown that. So no T cells will be forming the plasma cells, we can say.
No, not at all.
Okay, ma'am. So, ma'am, the differentiation of that two will be forming plasma cell and memory cells.
See, we have, so not today, but we did talk about the effector cells that come from T cells. So the T cells can either be based on the T cell receptor, the CD molecules present with it. They can either be the CD4 plus or CD8 plus. The CD4 plus molecules, when they are activated, they form the effector T helper cells, which can produce cytokines to activate some other cell types. But the CD8 positive T cells, upon activation, they become cytotoxic T lymphocytes. And these cytotoxic T lymphocytes, so these can either be of effector cytotoxic T lymphocyte or they can be of memory. So they can also be the memory type of cytotoxic T lymphocyte, which is not going to do anything, but is just a memory that is formed.
Okay, ma'am.
Okay. Okay. So I think we should stop here. We are going a little slow. Okay, we'll see how. So maybe I can take the schematics and have things written on it. So today's class was somewhat faster, but we still had more to cover. Okay, we'll see you in the next class then. Is there anything
Is it
from the
not possible
assignments?
that
Mm-hmm. Bolo?
no, ma'am, you can continue with what you are saying,
No,
then
no,
I will
I was just
continue.
saying that we'll see you in the next class, but if we still have more, we can go on.
Ma'am, when the antibodies are present on the B cells encounter the antigen, then only they will use the transmembrane protein and penetrate into the blood vessels.
See, one thing that I would like to uh, point out is whenever you're trying to say something in biology, we'll try to keep our terminologies very correct because some things can mean some change because it's not like they are losing the transmembrane domain. We'll come to that, right? So it's something else is happening there. So the transmembrane, for it to get secreted out, now the IgM molecule that is actually produced, it is lacking a transmembrane domain altogether. So let's not touch it right now itself. It might get confusing. Maybe we can do that instead of moving into the TCRs in our next class and then coming to antibody. we can first discuss the shape, structures, and types of antibodies so that this discussion from today is more meaningful to you all. We can do that, but we'll see what is happening.
Okay, ma'am, no problem. And ma'am, one last thing, I am not very much clear about the post capillary venue that you have shown in the diagram.
Mhm. See, over here, so one thing is your lymphatic vessels that is draining the lymph nodes, and the other thing is your blood supply. So the blood supply, so the blood supply can either be coming from the arterioles or the venules. So what is the arteriole? It's the capillary from which is having fresh blood, and venule is from is the capillary network of veins which carries your drained off blood, right? So one such example given here is the post-capillary venule. So the veins along with the blood, it also has your cells of the immune system, the lymphocytes. And so what is nice thing about capillaries, venules, arterioles is they are just one cell layer thick. So they're just one cell layer thick. The process of extravasation that we have previously read about, your lymphocytes can actually cross through this cellular barrier, which is the lining of the venule, and then they can come into the lymph node. So, so they have shown arteries and venules on one side and the lymphatic vessels on the other side. Just imagine that you have the venules from where we have the lymphocytes coming in. So this is where essentially, so this diagram is fitting in here. So we have the lymphocytes also here, and it can come into these spaces to have a very similar structure to what we see here. Is this clear?
Okay, ma'am, got that point.
Hmm. Okay. Anything more?
No, ma'am.
Okay. Okay, then we'll close the session and we'll try to fix this platform. Maybe you guys receive a new link for the next week. We'll see. Okay, I'll see you all in the next class.
Thank you so much, ma'am.
Thank you.
Thank you, ma'am.